So now let's go to our third research panel. It will be Nathaniel, Raviv, Jing Sima, Sid, Jane, Zhiying. And the topic will be DNA storage and systems. And you'll have 25 minutes. Clicker is here. The down button. Okay.
While the panel is getting ready in the spirit of Shuki, I wanted to start with a joke. And it was me about a week ago finally realizing that paradise stands for parallel and distributed. It has never occurred to me that this is the goal. But when I spoke with a few people here, I understood that I'm not the only one that haven't made that connection. So maybe a show of hands. If there anyone here that did not make this connection? Okay. And that was my next question, is maybe resolve it once and for good. Is that the reason the lab is called Paradise?
The reason it's called Paradise is that's how I felt when I came to Caltech. And then it relates to what Yuda said. You first make a decision, then you try to make a justification. so I think that's a heritage of Paradise.
Right, I'm glad we resolved this. Okay, I'm Nathaniel Raviv. I'm an assistant professor in Washington University in St. Louis. I was a postdoc here 2017 to 2019. And I had the pleasure to collaborate with Shuki and also with a few of the panelists here about an exciting, radical new technology of information storage in DNA molecules. I think this is one of Shuki's favorite topics. Maybe you can comment on that later. Maybe because it's like at the 45-degree angle between an imaginative, radical idea and new technical contributions that have to be made in information theory to realize that idea. So the idea of synthesizing and manipulating DNA molecules to store information did not start recently. It was conceptualized in the 1950s. Specifically, there's a saying that's attributed to Feynman that there's plenty of room at the bottom. If we want to store information, why won't we use nature's storage system, like Shuki says, and get a whole lot of benefits? Of course, miniaturization is an obvious benefit, right? You could fit the whole internet inside a fridge or something along those lines. Stability is also a huge benefit. DNA will be stable to tens of thousands of years, something that you can't say about your favorite SSD. But what my favorite is that DNA storage is future-proof, meaning as long as organic humans roam the earth, they'll have some interest in reading and writing DNA molecules, and the technology will never be lost, something that you can't say, for instance, about floppy disks. So we have a great panel here. We have Jin Sima and Siddharth Jain that were students here together with me, and Jing Wang that was preceded us by a few years. So the first panelist will be me. Each of us is going to mention a little bit about the work that they did while being here and forward-looking what other things they did since. So the reason I thought before 1 week ago that this place is called Paradise is because it so successfully combines research and friendships. Either one of them would not have been possible without the other. When I first came here, I started working on something that was completely new to me, about how to handle unordered packets of noisy information. What happens in DNA storage is that, imagine somebody takes information on your phone, chops it into small chunks of 1,000 bits, and puts them all in such a solution in an unordered way. How can you possibly manage an information system that behaves like this? More so, DNA is highly susceptible to mutations, and I'm going to quote Shuki again here. Shuki once said that if you want a proof that DNA accumulates mutations over time, just look at the mirror. I thought about that when I saw all the young and pretty faces in the poster in the entrance from 10 or 20 years ago. That's maybe one of the brutal truths that Shuki sometimes said. So our goal was to manage such an information system, specifically one that is resilient to noise. Me and also Jin here, we came up with this thing called the sliced channel. It's a channel in which the information is sliced into these fixed length chunks and put into a solution, and your goal is to extract the information successfully, even if some bits are being substituted or replaced or erased and so on. And the brilliant idea, which is mostly due to Jin, was a technique called robust indexing. It's a technique that enables you to attach an index, say 1 to x1, 2 to x2, in a way which A, is resilient to errors. Even if someone messes up with a few bits of the index, you still do not lose the order. And B, also, not only that it doesn't waste bits, right? Indexing is a waste of bits, right? These indices do not contain information. It enables you to encode some of the information into the index itself. So your information not only goes into the x1, x2, it also goes into the index of those xi's. When I saw this, I said, well, this has to be useful way, way beyond DNA storage. And also other ideas that we were talking about since. And after I got the position, one of my students came to me and told me, here there's this series of papers that are being published in all kinds of conferences for mechanical engineering about how to handle the security issue in 3D printing. Well, you all know what 3D printing is. You probably heard about ghost guns, like the assassination of recent CEO of United Healthcare. So there's a huge problem in the security world of how to tag 3D printed object with fingerprints, such that if a gun is being, say, 3D printed and then left at the crime scene, the fingerprint could somehow identify the perpetrator. And once my student told me that, I said, oh, that's very similar to what I did with Jin. So that's a similar math and similar ideas in a completely radically different application setting. So we came up with this thing called break resilient code. Now, the breaks are not a result of DNA synthesis. They're a result of some adversary that is doing this on purpose. And we, alongside with my first student and Jin, we developed this theory of how to extract information from an adversarially broken code word. And also, what I find very exciting in this project, that we actually did it. We took the printer, we implemented those codes, we printed stuff, we broke them. I think it's a wonderful experience for every information theorist to be able to hold a code word in your hand. All right, so I'm going to pass the mic to Jin. You want to come here? You can also sit. Sure, go ahead.
Hello, everyone. I'm Jin. I'm Shuki's PhD student, and I was here since 2016, and I left in 2022. So my thesis is about error correction in DNA storage. It's more about the theoretical aspects for dealing with noise that occur in DNA storage. So one interesting thing about DNA storage is that it's kind of different from traditional digital storage and communication systems, where I think, so in traditional, the error correction in traditional digital storage and communication systems have been well studied by classical information and coding theory. And there are some new aspects in DNA storage that was not quite well studied in digital storage systems. For example, one example is that in digital systems, you usually encode information into a sequence. You can write infinite, maybe not infinite, but you can write information in as long as you want. But in DNA storage, there is some kind of biological and technological constraints where you cannot simply write a long sequence into a DNA molecule sequence, into a DNA molecule. You can only write in short sequences. That's why we have this problem of how to correct errors when information is only carried or is only encoded into short sequences. That's the work we have with Nathaniel. I think Nathaniel had a very nice introduction, and the problem itself is, I think it's very general. It's not necessarily limited to the applications of DNA storage. I think that's the kind of the research style and the questions I think during my PhD, I feel that's partly Shuki's research style, that you look at first order problems that are fundamental. And actually, the other work I did was like the deletion and insertion error correcting codes. As Nathaniel mentioned, that there are a lot of mutations in DNA, and one of them is like the deletion and insertions in those DNA bases. And this work is about how to deal with those. Actually, this kind of problem also has applications in traditional communication like synchronization, and also has applications in bioinformation processing and natural language processing. So actually, I didn't know that my thesis was about DNA storage until my thesis candidacy. So because at the beginning, I think Shuki asked me to look at sequences and algorithms. These are very broad subjects. I think they kind of lay me the mathematical foundations needed to look at these problems. And then once you have those ingredients and sources, I think then you can cook anything you want, I guess. So yeah. And after my PhD graduation, I think I continued some of my work on DNA storage, but not related to, not necessarily error correction, but like DNA synthesis, where we look at how to use coding to do efficient DNA synthesis. And I also work with Nathaniel on how to kind of recover from breaks in 3D printing. And I think Nathaniel had a very great introduction. And also, I think another thing is like the lifelong curiosity about about the understanding of information stored in natural DNA. I think so. One treasure I had from after working with Shuki is the curiosity I had after graduation. I think it opened up my curiosity about many aspects, and I think that's and that's the thing I really value myself. Thank you.
I want to make one comment, Jean. Thank you. Jean is the most modest student or person I ever met, and I don't say it lightly. And I know he's embarrassed now, but I don't care. He solved in his thesis 3 major open problems, including one that people worked on deletion codes for more than 60 years. And he would just put a little line there. And he's also modest towards himself, which is very unusual, because typically when we work on a problem, he comes to my office and he's saying, I think I have a general idea how to think about it. What it means in Jean's language is that he solved it. So Jean won many awards for his work including the best thesis worldwide in information theory, and also the Caltech best thesis in EE. For me, it's a closure of circle because one of my mentors was Tom Cover at Stanford. He was on my thesis committee and really influenced the way I think about science. I copied from him. I'm now sharing a secret. I copied from him the model of how to do group meetings, where I have no idea what will happen when I go in, and he had the same system. That's the only rule. And this Best Thesis Award is actually named after Tom, that I think if he would be alive, he would be here today. So, Jean, I feel that it was a privilege to work with someone so modest, and I hope I would be able to increase your, remove an epsilon from your modesty. So people in this day and age where there is Twitter and Facebook and people say I'm honored and blah, 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 that they will recognize your talent because you are destined to make a huge impact on the world. So thank you.
Thank you, Shuki. Thank you very much.
So hi, I'm Sid. I worked with Shuki for almost 8 years. I did my PhD with him, and I also did I continued as a postdoc with him for 2 years. So today I'm going to talk about something a little bit different from DNA storage, which Nathaniel and Jin touched on. They touched on DNA storage that is kind of external, like it happens, DNA is stored in a solution, and then it's synthesized again, it's sequenced again, so there will be some errors, and that's kind of the errors they were touching. With Shuki, I kind of went a little bit of a different path, and the research question that we asked was, if we look at a single snapshot of DNA today, what can we say about its history? Now this question can go in both directions, like you can go backward, like the way the question is posed. So, or you can go forwards, like you start with some seed of life and how it evolves to get to some sequence that we see today. So I kind of worked on both the questions, and that led to some kind of different work in different applications. So if you think about the question where you have a seed of life and things evolve into multiple sequences through some mutational events, and in my case, I targeted specifically mutations that happen, which are called duplication mutations, or specifically tandem duplication mutations. The question was can we generate a large amount of sequences through one sequence, one seed, through tandem duplications, and if we can do that, can we do it for a large amount of seeds which are distinct from each other? So suddenly, if you can do it from large, by large I mean exponentially large, then you suddenly have a error correcting code, because it kind of, you have a large set, each of those individual seed is generating a large set of sequences. They are all distinct. They can be easily traced back to their seed. You have a code. So this has application in something you could think of, that if you have embed some information in a cell, the cell evolves over time, then the information will get corrupted, and you can trace back where it came from. So that was one of the major topics of my PhD thesis, which kind of for the first 4 or 5 years, my PhD thesis revolved around these questions. Then I took a more applied route, which is where it got very interesting. So Shuki will talk. I think Shuki has also worked in, like, been a startup person, and this is where I kind of entered a little bit of more of a startup mode, where now we have these big databases. Like, for example, when I was working with Shuki, there was a database called the Cancer Genome Atlas. Now, this database had about 5,000 genomic sequences from different individuals, considering like maybe 15 to 20 cancers were there. So we were like, I had not done that kind of data analysis. And these are very huge sequences. Each DNA sequence is 3 billion pairs, so it's like a huge thing. And there are 5,000 of these, and I have never done data analysis with this kind of big data set. And suddenly I'm working with Shuki, and there's this question that, oh, can we have some ideas that, oh, we can evolve sequences, we can generate sequences through these duplication mutations. Now we have these big sequences, and there are some repeat regions. Can we somehow get the history from these repeat regions? So because these repeat regions have both things, they have duplications, they also have point mutations. So if one point mutation happens, and that is propagated in multiple duplication events, you can say that only one point mutation has happened and not like 5 if there were 5 duplication events. So those kind of ideas suddenly we could use to get history of each sequence. Now we have this algorithm, but we have to like build it and build a system. And that, I think, was a very, very big challenge for me to start with, but it is just the nature of working with Shuki that you always are very optimistic, and you are like always curious, and you keep going and keep going. And at the end of the day, I was able to like contribute to building like a system which could say something about cancers from those sequences. And specifically for brain cancer or glioblastoma, we were able to make pretty decent predictions about the chances of getting that. So this was like my work with Shuki, and I was really proud of getting that experience of working with Shuki, because in my current work also, it's a little bit of, I work in a startup right now, and it's very, very similar to that kind of stuff that I was doing when I was with Shuki, especially on this cancer project, so that all the learnings I had, especially the attitude, was very beneficial. Yeah. Thanks.
Thank you, Sid. Sid is a super brave person. And the key that he was trying to figure out is, cancer happens to about 50% of the people in their lifetime. And so at least the concept that cancer is a disease is wrong. It's just part of life. And what we were trying to do is to see if we can look at the DNA of people and figure out what is the mutation rate and how their mutation pattern of a person is related to potential cancer that they might get later in life. And Sid was super successful in being able to identify the very high likelihood of developing a cancer before it happened that is currently not treatable. It's a glioblastoma. And that's a completely different framework for treating cancer, which is, let's treat it before it happens, not after it happened, because typically it's a problem. So the whole industry of cancer treatment is not focusing on prevention, but it's focusing on treating things that are many times too advanced. So thank you, Sid, for your being brave and creative. Thank you.
Hello, everyone. My name is Zhiying Wang. I'm right now with University of California, Irvine, so it's a short trip to here. When I was a PhD student with Shuki, I guess I was one of the first 2 female PhD students in Shuki's group, if that's correct. My topic during the PhD research was on coding for data storage, mainly for flash memories and for distributed storage systems. So I was not involved in research in the area of DNA storage. But by the time that I was about to leave Caltech, I knew that Shuki and some of the students and postdocs were working on different problems that are related to DNA storage. At the same time, being able to attend the group meetings with lots of postdocs and students from Eric's lab, I also knew that DNA is a fascinating media to do computation or to do information processing. So I was always interested in that area. So while I was thinking what to present here, I'm going to introduce a problem that has some relation to the research that was done here in Shuki's lab. So suppose that you have some image of a paradise image, for example, that you want to store using DNA sequences. So what you can do is basically to translate that image into binary bits, and every 2 bits you can represent by 1 DNA letter, ACGT, and then store that as some molecules inside DNA vessels. However, there is a drawback in the current technology, meaning that it's kind of expensive to store things using the synthesis and the sequencing process. So what you might want to try is, instead of having only 4 letters, ACGT, as a nucleotide, you try to create more letters. And that is something called composite DNA that was proposed a couple of years ago. So the idea is, instead of having those 4 letters, you use a probability distribution on those 4 letters. And as probabilities are continuous, you can have possibly infinite amount of bits in each position. So that's the basic idea. I'm not going to go into more details about it. But the idea is that when you store things, then maybe unexpected errors happen, then we may not be able to go back to paradise. So our image will be corrupted, and then we see an image of a normal beach, all right? Some other California beach, all right? So what can you do? So while I was thinking about this problem, how to model it and how to solve the corresponding problem, I thought of one of the PhD students that was kind of a senior academic brother in our lab, Yuval Kasuto's work on flash memories. And that work was joint work with Shuki, of course. And that work had a similar kind of pattern, but with, of course, a different setup. So based on that work, we kind of formulated the problem of DNA storage with composite letters and how to solve errors in that model, under that model. So I would say that the impact from Shuki's mentoring is it can be unexpected. So you never know that when things will come to your mind, like a particular paper you read that was written by Shuki, or a particular code that was given by Shuki. So I was one of the TAs of Shuki's IST 4 class. So before the quarter starts, Shuki gathered all the TAs and asked us, what do you think is the most important quality for a TA? What do you guys think? Come again? Ability to connect with students. So I would answer not to give the answers to the students immediately, because I tend to do so.
Be kind to the student.
So he said, be kind to the students. That was a little bit surprising for me, because I thought TAs should be helpful in the first place instead of being kind.
It's
and
amazing that you remember.
And that became something that I try to carry along as I continue to be a mentor and to be a teacher. So I'm really grateful to be a student of Shuki.
Thank you,
so much.
Jean. I just want to make a small comment. I want to thank you for joining my lab because you are the only PhD student I had. And I have two postdocs, two female postdocs. I know that for women, this is a very difficult very difficult career to build a family and to be in academia, and I congratulate and admire all the women that are able to do it, including you. I don't know how many kids you have now? 2. Wonderful. Thank you.
All right, thank you. Thank all the panelists. I would just only like to mention Eitan Yacobi, which was supposed to be here but can't for obvious reason. He's one of the world's greatest experts on DNA coding. We wish him and his family well. And that's it. Thank you for listening.